everyone. In this video, I'm going to talk about the notes in your notebook that cover Chapter 7, cell division, the cell cycle, and the division of the cells themselves. So in this first video, we're going to talk about how different life cycles use different modes of cell reproduction, including binary fission, and as an overview for mitosis, and how they produce genetically identical cells. We're also going to talk about how this cell cycle is controlled and why it's important to control whether or not a cell proceeds in the cell cycle. First, we're going to discuss asexual reproduction. In the case of asexual reproduction, the offspring are clones. Now, clones are genetically identical to the parent cell, and the difference between this and the process of mitosis is that the cells are alive, but they are not individual organisms. Asexual reproduction that produces clones, we're talking about organisms that will become their own sentient being. In the case of clones, any genetic variations are solely due to mutations, which are changes in the DNA due to environmental factors or copying errors. Anything that causes a mutation to happen is known as a mutagen, and these can be due to things like UV radiation or any type of chemical mutagen. But it's important to remember that mutations can occur spontaneously during the copying of the DNA itself. Now, back in Unit 2, we talked about how prokaryotes divide by a process known as binary fission, whereas eukaryotes can produce by a process known as mitosis and cytokinesis. Mitosis is specifically the splitting of the nucleus. There are eukaryotes that can reproduce by asexual means. One of the biggest misconceptions that students have going into this unit is the DNA of prokaryotic and eukaryotic cell. Now, the thing that students get confused with is the prokaryotic DNA. First of all, the fact that it is prokaryotic DNA. So what I'm going to do is I'm going to draw a small table just to help remind you of what we've talked about for these organisms. So the genetic information of prokaryotes and eukaryotes are both nucleic acids. That means that for both prokaryotes and eukaryotes, their genetic information is stored in the form of DNA. There is no difference here in the genetic information of these organisms. Another misconception that students have is the structure of that DNA. In both prokaryotes and eukaryotes, the DNA is double-stranded. In both of these, it is double-stranded, two strands held together by hydrogen bonds, just like we talked about in Unit 1. One thing that is different between prokaryotic and eukaryotic cells is the shape of the chromosome. So in the case of prokaryotes, they are circular, and in the eukaryotic cell, we have linear DNA, and it won't form the chromosome until mitosis. I think the part where students get confused is that the fact that prokaryotes will only have one single circle of double-stranded DNA. And so many times students think that prokaryotes have that RNA instead of DNA. That is not correct. They think that the DNA is single-stranded. That is not correct. It is still double-stranded. But they do remember the difference between circular and linear DNA. Now, in the case of sexual reproduction involves the fusion of gametes. It results in offspring with genetic variation. In other words, the genetic information of the offspring is going to be different than that of the genetic information of the parents. Gametes form by a process called meiosis. This is where the genetic information is going to be cut in half or reduced to half. Now, the DNA in eukaryotic cells is organized into very complex structures called chromosomes, and you'll frequently see me drawing them like this. Now, when cells divide, there are two types of cells. The first type is called somatic cells. These are body cells that are not specialized for reproduction. So they can include hair, skin, cells in the intestines. Any cell that is going to divide but is not a reproductive cell. Each somatic cell contains 2 sets of chromosomes that occur in what are known as homologous pairs. Now, with the homologous pairs, you always inherit one from your mother and one from your father. And with the homologous chromosomes, it's important that you understand that they encode for the same characteristic. So let's take, for example, eye color. On your mother's chromosome, you may have the allele or the DNA for green eye color, but on the father's chromosome, you inherit the color for brown eye color. So homologous chromosomes are chromosomes that encode for the same characteristic, but the allele itself may have variation. It may be different. So in this case, the characteristic I'm looking at is eye color. And you're going to inherit the characteristic of eye color, one from your mother, one from your father. But within that characteristic, there may be variation, such as green versus brown. When 2 chromosomes have the genetic information for the same characteristic, that is what is known as a homologous pair. Gametes have only one set of chromosomes, one homologue from each pair. They're what are known as haploid cells. They will contain half the number of chromosomes and therefore are designated as N. So in reproductive cells, each gamete is going to contain half the number of chromosomes, so that when fertilization occurs, the 2 gametes fuse to form a zygote that has a complete set of chromosome. And so the zygote itself is what is known as a diploid, because the number of chromosomes in the zygote is going to be 2n, or a complete set. So let's take humans, for example. The egg that eventually became you was haploid. It only had half the number of chromosomes needed for a human being. And in humans, the number is 46. Now, there's nothing special about the number 46. We share that number with potatoes. It's not the most amount, it's not the least amount, it's just the amount of chromosomes that we have. So an egg, which is haploid, would contain half the genetic information, so the haploid number would be N equals 23. Now, the sperm that reached the egg successfully and eventually became you is also a gamete and contains half the number of chromosomes. So when the 2 fused to become the zygote, or a fertilized egg that would eventually become you, these 23 chromosomes plus these 23 chromosomes become you as a zygote, as an organism with a diploid number of 46. So again, I'm taking these 23 chromosomes right here plus the 23 chromosomes from the sperm, and together that forms 46 chromosomes, which is the diploid number for humans. All sexual life cycles involve meiosis. Gametes may develop immediately after meiosis, or each haploid cell may develop in a haploid organism, the haploid stage life cycle, and eventually produces gametes by mitosis. Now, in the case of humans, we have a haploid cell that will never develop into its own organism. So we depend on fertilization to produce a zygote that has 2n for a baby to be born. But that's not the case for all organisms. Some organisms have a haploid life cycle, and the haploid cell becomes its own organism, so it never needs to be fertilized. But in the case of humans, that's not the case. There's always a zygote that needs to be formed from 2 haploid cells coming together. Now, in the case of eukaryotic cells, we divide by mitosis, Mitosis is very specific. It's the splitting of the nucleus, followed by cytokinesis, which is the splitting of the cytoplasm and the distribution of the organelles. Reproductive signals are usually related to the functions of the entire organism, not the environment of a single cell. So in other words, in the case of eukaryotic cells, there are hormones that control growth and trigger the division of cells. Most cells in a multicellular organism are specialized and do not divide. A common example of that would be neurons in a human. Now, the cell cycle is the period from one cell division to the next, divided into stages in eukaryotic cells. The M phase, which stands for mitosis, is the splitting of the chromosomes to form the two nuclei, followed by cytokinesis. Interphase is the when the cell nucleus is still visible and normal cell functions occur, including DNA replication. Interphase has three subphases: the G1, which is variable, meaning that it may last a long time, it may be a short time. The S phase, which is when the DNA is replicated, and G2 is when the cell prepares for mitosis, making all the necessary proteins, such as microtubules, for separating out the chromosomes. Now, this is one of those topics that I feel is best explained with a picture, so I'm going to show you the cell cycle and label these phases I just spoke about in the cell cycle. So this is interphase. The cell will spend most of its life in interphase, and then followed by that mitotic phase, the M phase, including cytokinesis. So let's talk about interphase and what happens in interphase, starting with the first part, which is G1. So this is where the cell is growing, doing its normal function, making proteins, synthesizing, everything that we have talked about this far. This is the normal growth of, or normal phase of a cell and what its function is supposed to be doing, including growing in size. Then follows the S phase, which is when DNA is copied. So the amount of DNA doubles in the S phase. Next is the G2 phase. This is the second gap phase, so it's still continuing to grow. Organelles are being replicated. There's processes that are getting ready for the next phase, which is the M phase. The M phase, mitosis, is broken up into prophase, metaphase, anaphase, telophase, and we'll talk about those details more next time. Followed by cytokinesis, which is the splitting of the cytoplasm. So here is a table summary of what we had talked about and what goes on in each of these phases. It is important to note that we will talk about the stages of mitosis at a later time. So these stages here that talk about mitosis will go into much detail later. But this does provide you some information about interphase and what is happening during these stages. So specialized cell functions mean that the cell will be making proteins that are specific to what the organ does. So for example, if the cell is a skin cell, then it's going to be producing things that are necessary for a skin cell to do its function. The other one that's on here is the cytokinesis, which is the cell separation or cell wall formation. This would be the cell separation of the membrane done by a cleavage furrow in animal cells, and in plants there is the formation of the cell wall. The cell cycle is highly regulated. It means that it is controlled. It doesn't just, the cell cycle doesn't run all the time. It's controlled when it happens. Experiments showed that the chemical substances could trigger phases of the cell cycle even if the cell was not ready for it. So basically they could take the chemical triggers or the chemical substances and inject them into another cell. So let's say you have cell A here. which is ready to divide. And so I'll just put an R for ready. And then another cell over here, which was not ready for division. And then you remove the chemical substances from this cell and injected it into this cell. And then this cell would immediately begin to divide, even if it wasn't ready. And so investigations were done to determine what the chemical triggers were. So those trigger substances turned out to be protein kinases. More specifically, one named cyclin-dependent kinases. Moving forward from here, I'm just going to call them CDKs. Now, you would have known that this is an enzyme because of the ending, the ASE at the end. But as the name tells you, this enzyme is reliant or depending on another protein called cyclin. So these CDKs catalyzes the phosphorylation. CDKs in general, kinases. What they do is they make phosphorylation of proteins happen. And in this particular case, this one is going to phosphorylate proteins that regulate the cell cycle and are activated by, and the cyclins themselves, the CDKs, are activated by binding to that cyclin. Now that cyclin is another molecule. It's kind of like, think of it like a protein helper. Sometimes students like to think of it as an allosteric regulator or a coenzyme. But what happens is the cyclin will bind to the CDK and together form this kind of globular structure that is known as MPF. MPF, maturation promoting factor. But it's only activated when these two are joined together. Now there's a variety of CDKs, and each of them has a cyclin that's going to activate to it, which is only made at the right time during the cell cycle. After the CDK acts, after the CDK is activated, that cyclin that bound to it begins to break down by a different protein. It's a protein specifically that targets, it's an enzyme that, and I know it's an enzyme, it's a protein enzyme that's going to attack specifically cyclin to break down the cyclin. And so that deactivates the CDK. So it's this cycle of going back and forth between activating and deactivating that CDK using the control of cyclin. So we're always going to have CDK ready and available. All we have to do is just dump in cyclin when we're ready for it to be activated, and then destroy the cyclin in order to deactivate the CDK. That way you're not making this all the time. You're just turning it on and turning it off. Cyclins are synthesized in response to various signals, such as growth factors. So these cyclins are not going to be just present. The amount of cyclin is going to change throughout the cell cycle, and it will change in response to substances such as growth factors. So I'm going to zoom in on one of the graphs that you have here and show kind of how these levels change throughout the cell cycle. So you have various substances that are shown on here, and the first of which is the cyclin itself. So the cyclin is represented by this line, and at the top you have the phases of the cell cycle themselves. And so you have, for example, the cyclin is going to reach its peak just at the start of the M phase, and then the cyclin levels will drop down as soon as M phase is over. Then what happens is the cyclin levels stay relatively low, and then just before the next M phase. So this part right here would be interphase, which is G1, S, G2. The amount of cyclin starts to increase again in the next cell cycle, reaching its maximum towards the end of the G2. and then the levels begin to drop off again because that cyclin gets destroyed. Well, as more and more cyclin is being produced, what you're going to see is that more and more of it are binding to the CDK. So the CDK levels remain relatively constant. But as more and more CDK binds to that cyclin, the two join, forming that MPF, and the amount of MPF begins to increase in the cell until again, it reaches a threshold. That's the trigger for the M phase. And then the amount starts to drop again because at this point right here, the cyclin is broken down. That says down. Now this shows you the same process, but it's showing you the CDKs and cyclin joining together. So you have the amount of CDK cyclin together, which is known as MPF, at its highest, and that is the trigger for the M phase. So the cyclin, which is this portion up here, is broken down, and what's left is this broken cyclin and that CDK all by itself. So that is now the start of G1. And as the cell continues through the rest of the cycle, so this would be S, this would be G2, the amount of cyclin is starting to increase again. The cyclin is starting to bind more and more to the CDKs, forming that MPF. And when the levels of MPF get high enough, that triggers the next M phase. Now, these CDKs function as cell cycle checkpoints. The G1, so there's 3 of them. The G1 checks to see if there's enough space for the cell, there are enough resources for the cell, and the next phase of the next resources for the cell and the next phase of the cell cycle, and it checks to see if the cell's even big enough to divide. DNA damage will always be assessed at every single checkpoint. But the first G1 checkpoint, there's 3 checkpoints in the cell cycle, checks for these specific things. At the G2 checkpoint, which is the next one, the cell cycle checks to see if the DNA has finished replicating, checks to see if the DNA was replicated correctly, and checks to make sure that the cell has all the material needed for the next phase of the cell cycle. Now this one has a typo on it. It should say the M checkpoint, not the S checkpoint. So please make sure you make that change in your notes. The M checkpoint checks to make sure that the chromosomes will split correctly, which means that they are all attached to spindles and will be equally distributed to the daughter cells. Now remember back from 9th grade, you talk about a phase in mitosis called metaphase, where the chromosomes line up at the metaphase plate. They line up at the center. And they do that because of these spindles, these proteins, that kind of play tug of war, to make sure that these all line up in the middle and that they are all each attached to wherever it is that they need to be attached. So the M checkpoint checks to make sure that they are going to be evenly distributed when these shorten and the spindle fibers pull these apart. Failure to pass each checkpoint will result in specific consequences based on that checkpoint. Okay, so if on an FRQ you're asked about that, please make sure you're giving a response that's specific to that checkpoint. Eventually, this uncontrolled cell growth can lead to cancer, but many times when you have an FRQ, if they're asking about a specific checkpoint, simply saying that the cell will become cancerous is not going to be sufficient. We want answers that are specific to that checkpoint that we're asking about. In the case of cancer cells, they do not respond normally to the body's control mechanisms. They can form tumors, and benign tumors are not harmful, or they can form malignant tumors which invade the surrounding tissues and can metastasize, meaning that they leave their location and go somewhere else. This is the end of part 1 for chapter 7 on cell reproduction. Thanks for watching. Bye.